Thank you very much, Tara, for that nice introduction, and it's a pleasure to be here. I very much enjoy speaking to teachers. I think it's a very important mission you and I have, and hopefully this lecture will give you a perspective on how we can teach evolutionary concepts, some of the exciting research that's going on in my labs and groups across the world. I titled my talk DNA from Archaeopteryx to Albatross, and this is a picture of Archaeopteryx, the first bird, and we'll sort of take a long tour through how scientists such as myself have used DNA to study the evolution of birds. It's a fascinating story that's still unraveling. And of course it begins with Charles Darwin. He was very much interested in birds himself, and although, as you may know, a little trivia, in The Origin of Species, his famous book published in 1859, he actually barely mentions birds at all, and birds on islands, such as the Darwin's finches that he's so famous for, he barely doesn't even mention. And so this, the fame of Darwin's finches has come about fortuitously, even though they weren't really mentioned in the origin. But I had the opportunity to visit his house in Down, the town of Down, which is about 16 miles south of London. It's a wonderful day trip if you ever happen to be in London. It's a big house. For many decades after the Darwin family moved out, it was basically a school, and in fact was becoming quite run down until recently, till a government tourist agency took it over and renovated it, and now it really is quite an attractive exhibit. You can walk into his study and see his copy of Karl Marx's, Das Kapital, whatever. He had a huge library. All his original books are there, and some really exciting exhibits for kids as well as adults. So it's highly recommended. Of course, I came there to soak up some of the intellectual heritage that he had left, and here's a picture of me soaking up that heritage. Deep in thought, I'm traversing what's called the Sand Walk, which was the little walk that Darwin took every afternoon after he had been writing for many hours and had lunch. He would take a walk around his property, sort of a sandy pathway, and think up yet further deep thoughts about evolution. But Darwin is often credited with introducing the idea of a genealogical tree, the idea that species that we see today have all descended from common ancestors. And this is a picture, albeit a backwards picture, but showing his conception of species going back through geological time. These are the geological strata. This was published in The Origin of Species. And you can see, however, that there's quite a bit of equivocation here. He's not really definite about anything. In fact, these aren't real species. They're all hypothetical. They have numbers and letters to designate them. You can see that these lines are all mostly dotted lines, and they're not all even connected to each other. Basically a very non-committal approach to what we call phylogeny today. And these days, whereas Darwin primarily used the sizes and the shapes and the behaviors of animals, we're now able to use the detail we can get from actual DNA and the chromosomes. And now our genealogical trees are much more precise. There's basically a lot more accountability on these trees. We have to back up all our statements with statistical tests. You can see some of the numbers on this tree here are indications of our confidence in particular branches. As the number gets close to 100, that means we have high confidence in the branching pattern. But a number like 57 means that essentially the relationships between all these species above it are somewhat in question. So there's quite a lot of advantages that DNA offers for such testing and for really making precise statements about the pattern of ancestry and descent. And this science of phylogeny or systematics is actually a very old science. It's in many ways the oldest science. Aristotle was a systematist insofar as he cataloged the biodiversity in his area. And the major tool of systematics, of phylogenetics, is the phylogenetic tree or genealogical tree. It describes the pattern of ancestry and descent, designating common ancestors and descendants through time. What can we use phylogenies for? Well, essentially, they're a roadmap to biodiversity. They give scientists a tool for organizing the bewildering diversity in nature and for drawing connections between the different species. These connections serve as roadmaps, as signposts for not only understanding how the species themselves are organized, but also for biogeography, which is the study of where species are found, the geography of biodiversity. It's also a useful tool for understanding the process of evolution, the speed of evolution, the pace of evolution. All of these can be very well quantified by using these phylogenies. And I hope today you'll get a feeling for some of the different ways in which we can use phylogenies to study biodiversity. Not just of DNA, of course, but of behavior and ecology, as well as genetics. So today we'll be talking first about deep time phylogeny of birds and some of the ancestry of birds and some of the evidence for the dinosaur origins of birds. We'll talk a little bit about a field known as bioinformatics and how that has shed light on the living species that are related to birds and how they're related to what the controversies are in the closest relatives of birds. We'll also talk about the time scale of avian evolution. Is it a recent explosion of species, or is it an ancient lineage of species? And then we'll end by talking about some of the tips of the genealogical tree for birds, in particular the songbirds, as well as the tips even extending into individual species. And the goal of phylogeny in the broad sense is to really understand this broad tree of life, to understand the entire genealogy and all its implications from very deep roots to the very twigs and branches. So Archaeopteryx, there it is credited as being the first bird. It was discovered in 1865, just a few years after the origin of species. And this was a godsend for Darwin insofar as there were many skeptics about evolution. One of the big problems with the theory that Darwin clearly recognized was that there are gaps in the fossil record and that we don't have a good record of the transitions that species undergo. Archaeopteryx was a perfect transitional species because it showed characteristics both of reptile ancestors as well as modern birds. For example, Archaeopteryx had teeth, which modern birds do not have. It had a very long bony tail, such as you see here. Many modern birds have quite a long tail, but it's supported not by a series of vertebrae, essentially, that are forming a bony tail. Modern birds have all their tail feathers embedded in a structure called the pygostyle, the big muscular structure at the base of the sacrum. And Archaeopteryx also had these unfused hands. You can see clearly that they're independently moving fingers on Archaeopteryx, whereas modern birds have forsaken that movement for creating a rigid surface on which their wings, their feathers can be embedded. And so modern birds have fused many of these elements in the fingers and digits. But on the other hand, there are a lot of bird-like features. And so, for example, the very light, thin skeleton of Archaeopteryx, the very long hind limbs, for example, and forelimbs is very reminiscent of modern birds. Of course, the feathers are a clear indication of genealogical relatedness to birds. There's a lot of controversies about where Archaeopteryx spent its time. Here's a depiction of Archaeopteryx in a fern frond, extinct fern frond. This is a frontispiece of a very famous monograph published in the 1920s by a German biologist. And clearly he's making the statement here that he believed Archaeopteryx spent much of its time in trees, and that flight may have begun as a gliding exercise from the trees down. That's one major theory for the origin of flight. Another theory, which is many people feel has better support, maintains that Archaeopteryx actually began flight from the ground up, and that it actually was primarily a terrestrial, albeit highly cursorial, dinosaur. That it spent a lot of time running, catching its prey, making short leaps perhaps, using those long forelimbs to capture prey items, and that led to flight. This is the kind of controversy that's destined to keep paleontologists gainfully employed for centuries, since we'll never really know the answer. But there's been interesting evidence on both sides. One of the recent spectacular findings in the last decade has been the discovery of a huge diversity of feathered dinosaurs. These are clearly dinosaurs and related to a variety of groups such as theropod dinosaurs, which is the group to which Tyrannosaurus rex is affiliated. And yet they are clearly non-flying. There's nothing about the morphology of this species, which is actually an unnamed species in a group called dromaeosaurs. There's nothing in this species that suggests flight. It doesn't have the long types of feathers, and especially on the primaries and secondaries of the wing, that Archaeopteryx has. It only has a sort of a fuzz over much of the body and some feather extensions in the integument of the hind limbs as well as the tail. And so it's very intriguing, therefore, to ask, well, why did feathers evolve in the first place? They don't seem to have evolved initially for flight. Perhaps they evolved for energy conservation or temperature maintenance of a warm body temperature, or perhaps they were involved with signaling and communication, and that they were brightly colored as they are in many modern birds. So the exquisite preservation, though, of these fossils has really filled in many important gaps in our understanding of avian evolution. And this just shows a series of dinosaur species leading to Archaeopteryx and other birds. These are modern birds here. This series includes a very interesting species called Microraptor, which actually had fairly well-developed wing feathers on both its forelimbs and its hind limbs. And the claim was made, actually, that in fact flight went through a four-winged stage during dinosaur evolution. Now, I'm not sure I'm really willing to buy that particularly, and that's partly because of the placement of Microraptor in the genealogical tree. Had avian evolution undergone a four-winged stage, a genuinely four-winged stage, we would predict that Microraptor would be confidently placed somewhere along the line actually leading to birds. Because it's placed up here on a subsidiary branch, it's conceivable, but it's by no means clear that this four-winged stage was present on the line leading to modern birds. What we are confident of, though, is the many features shared by all of these dinosaurs that are in common and that we can faithfully trace to these ancestors, as you might say, leading to this branch. And these are, for example, the cursorial mode of life. All of these species are highly cursorial, and many of them are predatory. Many aspects of the hand morphology and the foot morphology are in common and can be traced to this branch. And so again, this is a simple example of how we'd use genealogical trees to understand the sequence of character change, the sequence of the shapes and morphologies of ancestors of birds. Now, modern systematics has largely, although not completely, turned to DNA for its evidence. And it's interesting to ask why this is the case. I mean, I'd like to think that in some way DNA holds a sort of unbiased record of the genealogical tree. I think that's certainly true. Some people have argued that in some way DNA is more central to sort of the essence of organisms and their development. I think DNA has a very important role to play in development, a crucial role, although it's debated as to how whether or not DNA is actually essential. And so, for example, paleontologists will take the view that morphological traits are equally good at recovering phylogeny as DNA. And the debate has simply never abated. But to give you an idea of how we work with these resources, traditional museum specimens, such as you'll see after this talk, are organized in trays, and they're a great record of the phenotype or the outward appearance of species. What molecular systematists such as myself will use are not so much these types of specimens, but the small bits of blood and tissue that we're able to collect from living species. And many museums across the country are actively engaged in creating libraries of species across the globe. In fact, there's an effort right now called the Barcode of Life to assign a short DNA sequence, a short but informative DNA sequence, to every species of bird on the planet. That's about 9,800 species, almost 10,000 species. And this barcoding effort is very much a worldwide effort that could have a lot of practical uses, such as being able to identify species from very minute pieces of information, such as scraps of feathers, a little bit of skin or blood, lots of applications in forensics, lots of applications in terms of figuring out strikes of airplanes at airports and things like that. So our libraries may not be as visually exciting as those from the traditional specimens, but they have a lot of information from them. We actually can get DNA from the traditional specimens as well, and I'll talk a little bit about that towards the end. Now, we use two major types of tools in our study of phylogeny. One is from the mitochondria here. It's actually the most commonly used tool. It's called mitochondrial DNA. It's partly commonly used because it's so abundant. In every cell, there's probably on the order of several hundred copies of a mitochondrial DNA. It also has a number of interesting advantages. It's small, happens to be circular. It turns out it's maternally inherited, and so even in mammals, inheritance is strictly through the maternal line. So all of us in the room here have inherited our mitochondrial DNA from our mothers. It turns out that despite its crucial role in energy metabolism and specifically in the electron transport chain, all of these genes are encoding proteins that are facilitating energy exchange in our cells. It turns out that despite this crucial role, it changes very fast. A typical prediction in evolutionary theory is that very important structures tend to change slowly. And mitochondrial DNA clearly violates this paradigm, partly because many of the changes in its proteins, in its DNA sequences, turn out to be what we call silent changes. They don't actually change the sequence of the proteins of the amino acids that they're encoding. They occur in certain positions that, although they change all the time, the amino acid sequence is preserved, although not completely. So nuclear DNA is the other tool that's frequently used. It's completely the opposite of mitochondrial DNA. It's large and linear, so mitochondrial DNA is only about 15 or 16,000 base pairs. Of course, all the DNA in our genomes, the human genome, for example, is 3 billion base pairs, so much, much bigger. It's inherited from both our parents, of course. It shows a much slower rate of evolution. And it has this habit of recombining, meaning that the copy from our mother and the copy from our father will cross over occasionally. And you can imagine how this would make it very challenging to disentangle the sort of linear thread that we can trace through DNA trees. Becomes very complicated, particularly when we're studying very closely related individuals and species. But we'll show you some examples of each of these and their respective advantages and disadvantages. So we're going to talk about several different scales, very broad scale phylogeny, as I mentioned earlier. We'll have an example of how mid-scale phylogeny can tell us about behavior and ecology. And then we'll end by talking about sort of fine-scale differences between individuals and populations. Now, to begin the broad-scale study, we saw how fossils played a very important role in understanding avian evolution, but we can't actually get very detailed DNA information from fossils. The oldest fossils we can reliably get DNA from are really only a few 100,000 years old. And of course, the fossils such as Archaeopteryx and other dinosaurs are many tens of millions of years old. So we turn to modern species to get very detailed information about their DNA patterns and structures. And this just shows a particular tool that I've been involved with called a back library. It's a library which consists of parts of chromosomes, essentially. Basically, each of these parts is a few 100,000 letters of DNA in length. So, which is quite a lot, actually. 100,000 base pairs, 200,000 base pairs is quite a lot of genetic information. And these back libraries allow us to harness that information and organize it so that we can basically point to any particular position on the chromosomes and study that particular gene. Just makes it much, much easier. And you can see that we've directed our efforts to studying the ancestors of birds, such as these reptiles, turtles, Tuatara, which is this ancient lineage from New Zealand. You can see it has a very big genome. These are the total size measure of the size of all the DNA in the chromosomes. You can see it's quite a bit bigger than the human genome, even. A Gila monster, which is a poisonous species of lizard in the southwestern US. A garter snake, an American alligator, and some very primitive birds, such as emus, which you can see here, and here's a picture of one. It's found in Australia. So by studying these ancestors of birds but still living, we hope to get some very detailed information on how bird chromosomes are arranged and how they've changed. And this is just not necessarily important, but just all the many, many uses that these particular resources called back libraries can be put to. They're useful for studying phylogeny, also for studying development and how organisms go from a single cell to a multi-cell, and many different aspects of evolution. So we did an interesting project where we kind of took a very broad brush view of the chromosomes of birds. We wanted to understand what a reptile genome looked like. And by genome, I simply mean the collection of all the DNA in the chromosomes. The bottom line is the chicken has been studied in a lot of detail. In fact, the chicken genome is now sequenced. It's about half the size of the human genome. There's also a species of bird called the zebra finch, also lives in Australia. Its genome is being sequenced. It turns out that that's a very important model for studying how animals learn, particularly how they learn vocally. Zebra finches learn their songs through vocal cues just like humans learn their speech. And so we're hoping to make some links between the chromosomes and learning in that way. But it turns out that most reptiles aren't the stuff of laboratory populations. You don't usually see experiments being done on turtles and alligators, and yet they form a crucial position in this genealogy. So we wanted to learn basic facts about what a reptile genome looks like so we can better compare it to birds. And to do that, we underwent what's called a back-end sequencing project, where we simply obtained a very short sequence at the ends of many, many thousands of these clones. So each of these sequences might be 500 base pairs, very short. And we basically multiplied them across many different clones, which presumably are spread throughout the different chromosomes of these species. So we're getting a very broad brush view of the global patterns. And we undertook an exercise where we undertook a simple count of some of the DNA patterns that we found in these sequences. And this is where we're getting a bit into bioinformatics, sort of using computers and DNA to understand patterns within genomes. And so this picture here shows what we call a genomic signature. It's sort of a window into the particular pattern of word usage of a species. And of course, by words here, I simply mean combinations of the 4 DNA letters. So here are examples of 2-letter words. And when you see a dark pixel, such as this CC or this GG, that means it's a very commonly used phrase or word in that particular species. When you see a very light pixel, such as the CG or the TA, that means it's a very rarely used word. And it's of interest to ask why there are these differences in the first place. What's special about CC that makes it so much more common than CG? And that's something we can get to. This shows, in fact, the same sequence that we analyzed in the first image, but now we've decomposed it into 5-letter words. And you can begin to see some very intriguing patterns emerging, sort of global patterns. So one pattern is that this diagonal here, you begin to see these diagonals. And these turn out to be words that consist only of, for example, A and G on this diagonal here, and C and T on this diagonal. We also see, again, this pattern in this region here, where words that have this, this double nucleotide CG tend to be very rare. And it turns out that CG is what we call a very mutagenic dinucleotide. Whenever it appears in the DNA sequence, it tends to change very quickly because of particular biases in how the sequence is replicated and how it's copied. So sort of its frequency at any given point in evolutionary time is going to be very, very low. And we can compare these frequencies across different species to understand the changes that have occurred in evolution. This shows a more detailed pattern. Now we're breaking it up into 8-letter words, and of course there are many more such words. And you can begin to see the very rich source of information that these kinds of analyses can describe. Many interesting patterns which are found at many different levels in evolution. For example, we know now that these cruciform patterns here are found primarily in vertebrates, mammals, birds, reptiles, fish. But invertebrates, such as fruit flies and other insects, don't have that pattern. So something's going on with the DNA in different lineages that causes these patterns to be different. Now, we undertook an exercise where we kind of put all of these words, we studied all these words in all of these reptile species that you can see here. And what I want you to appreciate here is that you've probably been exposed to the idea that when we compare different organisms or when we compare different DNA sequences, we often first take a step of figuring out what's homologous to what, right? We want to compare arm bones to arm bones, right? We want to compare a certain gene of one species to that same gene in another species. That's how we first establish homology, which is very important for understanding evolution. But here we've taken a completely different approach. Remember how we sampled these sequences from the genome. They're scattered all across the chromosomes. And chances are we haven't sampled the same places in the chromosomes of all of these different species. Rather, we've put all our information through this blender that we call genomic signatures. And we've asked, is there any information that is sort of over and above the position that we find them or the particular gene that we find them in these organisms? And the answer, surprisingly, is yes. This genealogical tree here is based strictly on the differences in the patterns that we see between these images. And what's remarkable is that, for example, the two mammals go together. That's exactly what evolution would say based on other data. The frog, Xenopus here, comes out near the base. That's exactly what other pieces of information would say. And chickens and alligators are quite closely related. So all of that is confirmatory with other pieces of evidence. And it tells us that these sort of word frequencies, which are spread across all the chromosomes, actually do contain some information about phylogeny. Now, the one surprise in this analysis was that the turtle here ended up very close to the chicken and the alligator. And most of the fossil record will tell us that that's not true, that in fact turtles should be much deeper in the genealogical tree for this group. They should be one of the first reptile lineages to branch off. But in fact, there have been a number of recent studies suggesting that, based on traditional phylogenetic analyses, albeit of molecular characters, suggesting in fact that turtles are quite closely related to birds and alligators, completely changing how we might view the change in the morphology across this tree. So we were actually surprised at how consistent our patterns were using this very unconventional approach compared to more traditional phylogenetic approaches. Now, one of the most comprehensive studies of the evolution of birds was done by a guy named Charles Sibley. He was a professor at Yale for a number of years, and then he moved to California. And he conducted probably the broadest survey of DNA evolution within the birds. He used a technique called DNA hybridization. And in this technique, we're not looking directly at the DNA, but rather we're taking the DNA of one species and sort of melting it together, the two strands, with the DNA of another species. We're making what are called heteroduplexes. These are double-stranded DNA molecules where one strand is from one species and the other strand is from a different species. And when we have these heteroduplexes, we can actually heat them up, and you can imagine that species that are very closely related will have very similar DNA patterns, and they'll take a lot of heat to melt. Whereas species that are very different from each other will have quite dissimilar DNA patterns, and they won't require as much heat to melt. So we can use this difference in temperature to create a measure of divergence. And that's exactly what Charles Sibley did. Quite a quantitative approach. You can attach numbers to all these things. And you can see the fascinating tempo of evolution in this group. For example, many of these branching points are extremely compressed in time. It's not as if we have long periods of evolution along a single lineage and then a branching event. Many of these branches in the interior of this tree are very, very short, almost as if there's been an explosion. And indeed, that's one of the thoughts, that there has been a genuine radiation of birds. The controversy is more when that radiation occurred. Like many techniques, Sibley showed that the flightless birds, such as ostriches and emus, were near the base of the tree, and that the songbirds and the perching birds, such as you see over here, these are thrushes, chickadees, most of this common species we see in the woods around Massachusetts and New England, are at the complete other end of the tree, much more recently evolved. Now, with regard to this timing issue, if you look at the fossil record of birds, and that's what you see right here, these are all the different orders of birds, the different major groups of birds, and these lines show the extent of fossils going back in time that we can assign confidently to these groups. So for example, though you can't read it, this says Psittaciformes, those are parrots. How old is the oldest parrot that we can unambiguously place in this modern group? Well, the answer is generally about 65 million years. That's the age that most paleontologists would say our modern birds have evolved. The DNA evidence gives a very different picture. The DNA evidence suggests, and I'll get to how they get to this suggestion, that in fact most of the major groups of birds diversified much closer to 100, perhaps 110 million years ago. And to get this, they use a concept called the molecular clock. Now, the molecular clock, as you can imagine, has this idea of gradual, fairly constant change in it. And basically, the idea is that the rate of change of DNA molecules is roughly proportional to time. Now, you can imagine all kinds of factors that might make that statement very untrue, that might throw a lot of noise into this clock. And suffice it to say, there are a variety of techniques that we can use to assess how equivalent the rates are in different lineages. In general, the rates seem to be fairly similar in different groups of birds, although some lineages of birds seem to be going faster than others. And so, for example, most songbirds have a generation time, that's the time from birth to first reproduction, that's very, very short, often just 1 year. Okay? That's the generation time of a typical songbird. By contrast, many flightless birds, such as ostriches, emus, take 5, 6, maybe 10 years for one generation. And it turns out that this generation time is one of the main factors influencing the rate of evolution. Short generation species having faster rates sort of on a per year basis. So the paleontologists have been very quick to take issue with this molecular clock. And of course, they make all kinds of jibes at us. They say that our clocks are unsteady and they're not trustworthy. And of course, we in return say that their fossil record is scrappy at best. So never the twain shall meet, although I have to say there is a growing amount of evidence which is causing the paleontologists to come over to the molecular system at this point of view. And so, for example, this slide shows a recent fossil found in Antarctica, okay? There are very few, there's certainly no, there's a very few terrestrial species that live in Antarctica. This is a species of duck that was very well aged at about 110 million years ago or so. And intriguingly, we can place this fossil duck on a genealogical tree for modern birds. Here are many living and extinct species of ducks. Here are some, there's a chicken, galliforms, and some ancient lineages such as the flightless birds. And what's intriguing is that by taking into account this very well-established branching order, right? No one argues about the fact that ducks and chickens and flightless birds are closely related. What's intriguing is that they can place this fossil right here and thereby constrain the ages of these deeper nodes. We can immediately say that these deeper common ancestors have to be older than 110 million years because we know that this aged fossil occurs at this point. So that's one technique by which the paleontologists are reconciling the fossil record with some of the molecular data. They're placing known aged fossils onto well-resolved phylogenies and constraining the more ancestral common ancestors. Now, that was the very deepest branches of the genealogical tree. If we look at the more recent branches, such as the songbirds, we see some very interesting patterns. Here are the major branches of songbirds. One group includes what are called the sub-Oscines. These are perching birds, very much like our chickadees. But for example, sub-Oscines typically do not learn their song. Suboceans have a very simple voice box. That voice box is called a syrinx. You can see it, yeah, there's syrinx right there. So a very simple syrinx. And it turns out that you can raise a subocene in isolation in a cage in captivity, and yet it will be able to produce a fully adult, mature song. You can't do that with the oceans here. Oceans are characterized by actually learning their song. They require communication and input from their parents or other individuals. They have a much more complex syrinx, many more muscle attachments to the syrinx, and their brains, in fact, have structures in them that are non-existent in the suboceans. So that's a major division within the perching birds, and even within the oceans, there are yet further divisions. The Corvida is a group that's confined primarily to the southern continents, Australia, New Guinea, but as its name implies, a sort of corvid root to its name, this is also the group that we think modern crows and jays originated. And so one theory, in fact, says that our crows and jays ultimately can trace their roots to the southern hemisphere, and that they entered North America through the Bering Strait, having come through Asia. This shows a slightly revised picture. You can see it's subtly changed from the previous diagram. In this diagram, the Corvida and the Passerida sort of have equal status. You can see they come to a single ancestor. We call these sister taxa. Sister taxa means they're each other's closest relatives, okay? So this was the result from DNA hybridization information, that Corvida and Passerida were sister taxa. More detailed information from actual DNA sequences suggests a slightly changed genealogy, but one that has some important consequences. Here you can see that there are many lineages of the Corvida, in red here, which are ancestral to Passerida. So now you can see they're not so much on equal status anymore. We've rotated the tree slightly such that Corvida now is branching off earlier. And although this may seem like a subtle change, it actually really nails down one of the most important conclusions from this work, and that is that many of the oceans actually originated in the southern hemisphere, not just crows and jays. In this slide, it's actually quite ambiguous where this ancestor would have lived. Maybe in Australia and New Guinea, or maybe in the New World. Whereas in this slightly changed phylogeny, it's very clear now that all of these lineages are southern hemisphere. And we can now say with confidence that the oceans originated in the southern hemisphere and dispersed throughout the world. And this picture here, published a few years ago, illustrates this idea where we have a Gondwanan origin for perching birds with a spread throughout many different parts of the world. Here are the lineages that came into the New World. So for example, our crows and jays would have come this way. Also things like thrushes would have entered North America from Asia and come all the way down to North America and South America. The other major branch, the suboceans, branched off quite a bit earlier with offshoots primarily in the New World tropics, with a few in the Old World tropics as well. So our picture of the spread of the biogeography of perching birds is becoming much more detailed now. I want to take you briefly on to show you how we can use phylogenies to study behavior and ecology. These are, again, some of my favorite birds. We can look at some of these specimens in the museum after this talk. These are New World jays, as I was just talking about. About 30 different species. And they are intriguing because they have very complex social behavior, or at least some lineages do. Pinion jays, which live in the Southwest, and scrub jays have a social system that we call cooperative breeding. It's a system wherein individuals will forsake their own breeding to help their parents, essentially. Even though an individual is physiologically capable of breeding, it will refrain from breeding and help his or her parents or some other individual. I mean, this is a real paradox. Natural selection says we shouldn't be doing that. Stellar's jays, by contrast, don't have a helping system. But they do have a somewhat permissive system in which they let other individuals come into their territories. And the question is, which came first? Many theories would predict a simple system, such as we see in Stellar's jays, would give rise to more complex systems, such as we see in scrub jays and pinion jays. How can we answer this? With a phylogeny. This shows the phylogenetic relationships of these and many other species of jays, with the cooperative breeders indicated in orange here. And what you can see, in fact, is that cooperative breeding, and in fact the very complex cooperative breeding that we find in pinion jays, is, if anything, the ancestral state, because these lineages are branching very near the base of this tree. So, for example, cooperative breeding has actually been detected in Old World jay species, such as Siberian jays. They're actually related to our Canada jays, which some of you may be familiar with, and also even in crows. So this habit of helping is very ancient within the New World jays and crows. And in fact, this more simple solitary state is what we call derived. It's more recent, having evolved from a more complex state. So this shows you, again, how phylogenies can provide this roadmap telling us the sequence of events in this case, behavioral evolution. And there's just the sequence there. So I want to just end with a few examples looking at the very tips of the genealogical tree for birds. This just shows some of our field work in Australia, where we do a lot of work looking at very recent events in evolution. This just shows some of the reception that we usually get. Usually we work in areas where there are very few people, and yet the Australians have this way of making themselves heard. But it's a very interesting place to work. This just shows some of our wanderings over the years. I'm not sure if there's any method to this madness, except perhaps the trips seem to be getting shorter and shorter, perhaps as my own family group gets larger and larger. I don't know. So this just shows, again, now we're looking at a genealogical tree that's very, very fine. So the tips on this tree are not families or whole species, but they're just individuals. In this case, we've used mitochondrial DNA to compare different individuals. And you can see how we can begin to infer where these individuals have moved. And so, for example, here I've color-coded these lineages based on where they were sampled in this landscape here. And you can see, for example, that here's a situation where these red lineages here are closely related to lineages in other populations, right? Here's a red lineages. These are birds that we sampled right here. They're nonetheless more closely related to individuals found in this blue location, right? There's that group there. Or in this yellow location, which is right here. This is not what you'd predict, but in fact, this is evidence of what we call gene flow. This is physical evidence of the movement of genes across the landscape. In this case, we can say at some point in the past, an individual from this red population moved essentially to the yellow population. Or maybe it had a series of birth events that successively moved in that direction. But you can begin to see how we can create very detailed scenarios of movement in biogeography by looking at this very fine scale. And this just shows a picture. That previous picture, again, was mitochondrial DNA. Fairly straightforward picture, actually. Now that we're moving into the nucleus, and we have hundreds of different genes we can look at, you can see how complex the patterns become. In fact, every gene here, these are all different genes, every gene is telling a slightly different story. And the challenge now is to figure out what's the single overarching story that can explain all these different complex patterns. I mean, we know that in evolution, there was one scenario, right? One sequence of events, however complicated that sequence may have been. And so the challenge is to figure out what that sequence of events is based on a plethora of new data and very complex patterns. Okay, just to give you an idea of what we can learn from those complex patterns, we can actually we can estimate the amount of genetic diversity within a population. So for example, these circles are an indication of how much genetic diversity is within a single location. This population here has quite a lot of diversity, whereas this one has very little diversity. And we can then, using some assumptions, we can actually translate this diversity into an actual estimate of the number of individuals in nature. I mean, that's really remarkable if you think about it. It's sort of like a remote sensing system. We can actually make some guesstimates of the rough number of breeding individuals in different populations. Not only that, but we can tell you roughly how these populations are connected to each other, sort of like we saw in the previous example from the babblers. Here are the arrows linking different populations with very thick arrows, such as here, indicating lots of movement between populations, and very thin arrows here indicating very isolated populations. They're not exchanging genes. And you can see the very complex scenarios that we can actually reconstruct from these DNA patterns. And it's very exciting to think about the detail with which we can probe this very recent evolutionary history. Okay, I'm just going to speed through a couple slides here at the end. I'm just going to let me just quick and see what else I have here, because I want to make sure I end with I don't want to go on too much longer. I'll just tell you briefly about a couple more stories that I think you'll find interesting. The first is a study of a very common bird called house finches. And we have actually been interested in understanding how they have interacted with this a bacterium, a pathogen called Mycoplasma. And this shows a map of how this Mycoplasma actually spread through house finch populations. Some of you may be familiar with the feeder watches that Cornell Lab of Ornithology runs. And it was through much of that information that this very detailed picture of the spread of this Mycoplasma came about. The birds get these lesions in their eyes, they have trouble breathing, and so it's very easy to trace this. And what we wanted to do was to ask how the bird was responding to the pathogen. And to do that, I won't go through all the details except to say that we were able to measure the gene expression of house finches when they've been infected with this pathogen. And this diagram here simply shows different genes that are either turned on when a bird gets infected or get turned off when a bird gets infected. And this sort of very detailed study of what we call gene expression, when genes are actually turned on, is going to be a very important use of DNA to study, again, very recent evolution. This infection of house finches took place only in the last 10 years, and we're very anxious to understand how the bird has evolved and how the pathogen has evolved in response to it. I want to just end by talking about, oops, this is just some extra stuff I don't have to bore you with. I want to tell you a little, just end with our recent study of Ivorybilled woodpeckers, since I'm sure many of you are as curious as I am as to whether they're really out there or not. This, of course, is the species that was rediscovered purportedly after not having been seen for 60 years. It was rediscovered in the swamps of southern Arkansas. And there's also been a more recent report of sightings in northern Florida. Very, very exciting if true. I'm not yet convinced, although I'm an optimist, and so I'm hoping that it is true. What we can do, though, is to study what ivory-billed woodpeckers were related to, and also we can help to build a database in case we should run across any feathers or feces, for example, in a nest hole. We want to know if that came from an ivory-billed or whether it just came from a pileated woodpecker. And so this just shows the, well, you can see this in a few minutes. We have the dubious distinction of having the largest collection of ivory-billed woodpeckers. Now, I first want to point out that I didn't collect any of these individuals. The second thing I do want to point out, and this is a little sobering, John Fitzpatrick, who was the leader of the Cornell group that rediscovered ivory-bills, he suggests that, in fact, ivory-bills may be the one species whose extinction was actually accelerated by scientific collecting. That's not something we're very proud of. There was a point in the late 19th century and early 20th century where every museum wanted their ivory-bill. And there was an awareness that the species was declining. It's unclear whether it's true, but I think if there is one candidate, it's likely to be the ivory-bill. Most other, in fact, all other North American species that have gone extinct have clearly gone extinct for reasons other than scientific collecting. But this just shows how similar ivory-bills are to pileateds. Although, I mean, at 100 yards, this would be challenging to tell apart. But with the DNA, we can do it quite easily. And this just shows a genealogical tree showing the relationships of ivory-billed woodpeckers here from North America to ivory-bills from Cuba, as well as comparisons to this imperial woodpecker, which is also extinct in Mexico, and many other species of woodpeckers, such as the pileated woodpecker. You can see how different the pileated and the ivory-billed are. They're nowhere close. So I actually maintain it's actually easier to tell pileateds from ivory-bills by their DNA than in the field. And the challenge is to get some concrete evidence of that. And we call this a barcode. This is what we're looking at for every species. It's through this sort of, hopefully you've seen a number of different examples of how we can use DNA from the very deepest branches of phylogeny to the very most recent tips, and can even use them to resurrect putatively extinct species. So thank you very much, and I look forward to your questions. Yeah, question in the back.
Why did you choose those birds in Australia?
Great question.
Like not South America or something?
That's a great question. What we tell them on our grant proposals, like why would you give us money to travel? Southern continents tend to have been much more stable in their environmental history, with the result that the biogeography patterns that we can find are probably going to be much simpler. And so, for example, Australia never had the extensive glaciers that we find in North America. In North America, 10,000 years ago, we were under a mile of ice, and now many species have rebounded northward as a result of the recession of the glaciers. That complicates our understanding of history. In Australia, by contrast, the change was much more gradual. What happened was there was rainforest about 15 million years ago across the entire continent. And then very gradually, over about 15 million years, the whole continent dried up, very much like that picture I showed. Now, this is a very dramatic change, but it's a very gradual change. And as a result, many species that are completely unrelated to each other nonetheless have very similar geographic patterns across the continent. And so we actually think that it's going to be easier for us to discern common themes in Australian biogeography than you would find in North America. They're simpler. Now, as to why we didn't work in South America, which also has some fairly simple themes, that's just serendipity. I happened to get a chance to go to Australia and New Guinea when I was in graduate school. So there's an element of determinism, but also an element of chance in it. Yep.
I'm not sure how to frame this question. If you're looking at mitochondrial DNA and then you have species whose gene pool becomes limited, say like with the condor,
Yeah.
how accurate then can your look backwards be?
Well, that's a great question. If we have species like the condor or the peregrine falcon, which, we essentially can't say anything about their natural history. What we can say something about is their human-influenced history. And in fact, many species that have been reintroduced into the wild show the signatures of having gone through a bottleneck or very nearly having gone extinct. They show very little genetic diversity. Many of their sequences that we see in different individuals are virtually identical. And that's really as deep as we can go. Essentially, when they're removed from the wild, much of their evolutionary history is erased.
In knowing that, then, can you also look back at a species that may have naturally gone through that same bottleneck
Yep.
of time?
Yep.
And see that same lack of diversity?
Absolutely. It gets more and more challenging as you move back in time, but we can actually make educated guesses about the size not only of current populations, like you saw in that one slide, but also the size of ancestral populations, using fairly complicated statistical tools and many, many different pieces of DNA. But there is something about a bottleneck which actually does erase some of the previous history. It becomes increasingly difficult to find out whether there was a period where the population was very big and had lots of genetic diversity. So
But can you do one more thing and then correspond that with natural catastrophic events that would have occurred at the same time?
absolutely. One of the things we're trying to do in Australia is to link our divergence events with environmental events. And so in the case of Australia, there's quite a lot of environmental change in this region up in the north here, right here, as well as here. And there's a lot of evidence from pollen, for example, showing that desert environments were becoming more prevalent at certain times. It's a little challenging because many of the divergences that we're estimating on the order of 600 or 700,000 years are actually quite a bit older than the environmental record goes. So it makes it more challenging. But in principle, that's exactly what we'd like to do. Yeah. Yeah.
If you can estimate the size of those ancestral populations, can you then use that information to predict the success rate of endangered species now based on their current populations?
Well, that's an interesting question. I mean, in general, there's this idea that genetic diversity is good. That it makes sense. Different genotypes will respond to the environment differently, and one of those genotypes has a better chance of surviving in the case of some calamity. And so, for example, with the house finches, one of the things we're asking is, do certain individuals that are more genetically diverse, are they more resistant, perhaps, than other individuals? But in terms of, and I think that's a valid sort of proxy. In other words, using genetic diversity for population health or individual health. Sometimes it becomes unclear how correlated those two things are. I mean, for example, there are a couple instances where species have rebounded very successfully over large geographic areas without very much detectable genetic diversity at all. And so, for example, elephant seals are one example of that. Also, it turns out that beavers in northern Europe have recolonized many areas where they were previously extinct, but have done so with virtually no genetic diversity in their populations. So it's a trade-off. It's never clear as to, maybe we're not looking at the right genes. Maybe they are healthy at other genes and we're just looking at the wrong ones. So I think there was a question here.
How much Ivorybill DNA do you have? Can you get any estimate from that about how big the Ivorybill populations were? And how confident are you of where they are on the woodpecker phylogenetics?
Right.
I just want to know because the kids are going to love that.
Yeah. We can get a reasonable amount of DNA. We typically take it from the base of the feet. They have quite a lot of meat in them, and it also doesn't compromise the specimen that much. Typically, we'll study mitochondrial DNA because it's easier. Although I suspect we could get some information from nuclear genes. We haven't done a population survey of ivorybills. It would be a fascinating thing to do. Maybe one of you could join the lab for a summer and do that study. Because that would tell you a lot about how genetically diverse they were. We've really only looked at a few individuals so far. And in terms of the confidence on the tree, we're fairly confident, although it's not 100%. So you can see these numbers up here. These numbers, remember, when they're 100, that's a good thing. So we're confident of sort of what this really says is we're confident that all these individuals above it form a group. It doesn't say we know what the branching order within that group is, but it says they form a group. Now these lineages here, we're not very confident of. You can see they're kind of drawn in a non-committal fashion. But we're confident enough to, there's actually, it's funny, in the final paper, which just was published, I can get copies if you're interested, it actually was unclear whether North American ivorybills were more closely related to imperial woodpeckers in Mexico or to Cuban ivorybills. So that was actually equivocal. And we have some ideas as to why that might be. We haven't resolved it yet. So.
How many Ivorybills do you have at Harvard? You want to say?
There's about 58, I think. So. Yeah.
Over
Was there,
how much time they collected?
sorry?
Over how much time were they collected?
Just a few decades. Most are from like the 1890s. And there's some as recently, I think, as early 1930s, perhaps. Yes?
Your premise is that if you have high biodiversity within the species, then your population must be large.
Right.
Well, sexual selection would select for a certain phenotype.
Right.
And you could have a large population, but the birds could all be very similar.
That's right. That's very perceptive, yeah. So the idea is that if you have some selection event, or maybe the females are all choosing a particular type of male, that might some way erode genetic diversity. It might, if suddenly one type is advantageous, it would sweep through the population and everyone would be the same. Well, that's exactly right. And in fact, one of the paradoxes that we're wondering is, okay, here we have closely related species that differ very strongly in their behavior, in their vocalizations, in their plumage, and yet their DNA is very mixed up. It's almost hard to distinguish them in terms of the DNA, the genes that we're looking at. And what that tells us is that selection may well have caused some genes to diverge very fast and to become fixed, or very basically gone to dominate the population very quickly. If we could actually find those genes, we probably would find there's very little diversity in the population. We're, of course, looking at genes that might not necessarily be linked directly to phenotypes, but that's a very active area of research, and people are actually finding those genes and looking at them in natural populations. So, yep.
When you're in the field collecting the cells to get the DNA, do you get it off the feet of the birds, and are they live specimens?
Well, we sample destructively, as it's called, and also non-destructively. It depends on the situation, on the species. Obviously, we don't get permission to sample destructively endangered species, and we wouldn't want to do that anyway. Typically, if we're sampling non-destructively, we'll catch them in something like a mist net, and we will take a small blood sample. Less frequently, we'll take a feather, and of course, we'll take lots of measurements of the specimen and then let it go. Actually, from a single drop of blood of birds have this distinction of having their red blood cells actually have nuclei in them. They have DNA. So from a single drop of blood, we can get a huge amount of information. For many studies, we actually would like to have the specimen, and also it's nice to have the specimen as a record, really, of environmental change. I mean, there's so many different ways now to use specimens. People are looking at chemistry and these things called stable isotopes, which record where the birds lived and what sort of environments they were in. And so I think there's a good justification for continuation of responsible collecting. But there we'll either catch them in nets or occasionally use firearms to capture them. Of course, species that are way high up in the canopy, it's impossible to catch by other means anyway. So, yeah.
During your fossil, in the beginning, when you were discussing fossils, you said that the dinosaur, some of them had feathers. How can you differentiate from the fossils, whether it's feathers or fur?
Well, that's a great question. There's a bit of controversy as to what these proto-feathers, as they're called, actually look like. And as you saw, some of them do have quite a fur-like appearance. And the key thing that people want to look for is this evidence of a rachis, which is this sort of central shaft. And actually, many of the proto-feathers have a discernible rachis. What they don't seem to have is the sort of finer barb and barbule structure that you see on modern feathers. Modern feathers have this very interlocking set of where they can form kind of a uniform surface. And we don't see that in some of these proto-feathers. And there have been a number of feathers in the fossil record that many people would contest. They just say, well, these aren't feathers. We don't know what they are, but they're not feathers. And so I think most people are satisfied, especially with feathers such as Archaeopteryx. I mean, those are essentially modern type feathers. They're not, there's minute details that are very similar to modern birds. Although some, such as the non-flying dinosaurs, it's debatable, although most people are comfortable with the idea. Yeah.
You're not really convinced about these sightings of the Ivorybilled woodpecker. Who exactly saw these woodpeckers? Bird watchers, scientists? Is the Cornell lab more convinced? Or? Right.
Well, I'll tell you, if I don't know. When it comes to bird watching, I would tend to trust bird watchers more than scientists. Well, it was a large team of individuals. I mean, there's a number of people now who are on record in print as having seen this bird in the last several years, either in Arkansas or in Florida. It's a tough situation. There's a lot of remote sensing going on. People are putting out automated recording devices and video cameras, trying to catch a glimpse of these things. There's quite a lot of circumstantial evidence of some of the, not so much vocalizations, but the pattern of knocking on wood. They have this very distinctive double knock, which isn't found in other woodpeckers. And there's some, particularly from the Florida group, some good evidence for those sounds. But yeah, it's very frustrating, obviously. I mean, there's a lot of ongoing effort now by teams of professionals and amateurs just trying to get what you really need is a photograph of one of these things, or a feather or some feces. That to me would be evidence enough if you could get DNA from it. Yeah.
Has anybody found any genes that are tied in or correlated with vocalization of songbirds?
That's a great question. This zebra finch that I mentioned, it's a small songbird, sort of the size of a sparrow, and they're commonly found in pet stores. I mean, they're hoping to unravel the whole song learning process through the chromosomes, through the DNA patterns. And there are a number of genes that have now been shown to increase in their expression in the brain when a bird hears a song from its own species. And these are very interesting because they probably are implicated somehow in the song learning process. It's a really remarkable translation of a wave pattern in sound to a physical pattern in the genes. And there's a bunch of candidate genes out there that play some role, although we still don't know how those genes work together. And so, we're hoping that having all the parts, namely the entire genome and all the genes, will give us a hint as to how they work together. Yeah.
There's nothing to do with biology. There's a beautiful flute solo by Debussy called Syrinx.
Oh, really? Called Syrinx? Oh, that's interesting. He must have been an ornithologist or been interested in birds. Okay. Huh. Interesting.
And I can't play Syrinx.
Thank you for that. Yes?
Is there any estimate whether the pileated woodpecker or the Ivorybill West had a common ancestor?
Yeah. Well, the pileated in Ivorybill, that's going to be very deep. That ancestor will be right down here. And I don't know offhand. It's probably 15, 20 million years, something like that. These guys had a much more recent ancestor, maybe on the order of 1 million years.
Thank you.
Yeah.